All right. So hello, everyone. Thank you for joining to the EUSynBioSeminar. And today we have Rahmi Lale speaking. And just to introduce himself a little bit, Rahmi Lale is an academic at the Department of Biotechnology and Food Science at the Norwegian University of Science and Technology, NTNU, in Trondheim, Norway. He is also the co-founder and CEO of Syngens, at an NTNU spin-off operating at the intersection of artificial intelligence and synthetic biology. At NTNU, he leads an interdisciplinary lab that integrates biophysics, computational, and synthetic biology. His primary research focus is on understanding and controlling transcriptional and translational regulation in various microorganisms, ranging from bacteria to higher eukaryotes. At Syngens, he leverages the predictive power of AI to engineer biological systems for biomanufacturing applications. So with that introduction, Rahmi, thank you very much for joining and for giving this seminar. So I guess the floor is yours.
Well, thank you very much for the introduction. Hello, everyone. So the setup is that I'm going to have a presentation about 20 minutes. So please hold on your laces and then you can ask the question at the end. So the title is Engineering Promoters and 5' UTRs. And it's a bit of a long story starting from 2016 up to now. So I will take you through how we started with the different engineering approaches we have on the regulatory sequences and all the way to the predictive DNA design. I think I skipped introduction. That was a brief and a nice intro introducing both the university, which is a Norwegian University of Science and Tech in town, where I am in Trondheim, Norway. And also I have a lab that focuses on biophysics, computation, and synthetic biology. So among the other things that we do is focusing on transcription and translation regulation in different microorganisms. So one of the unique angles that we have in our lab is that we are not limited to a certain model. We work with many multiple organisms. But irrespective of that, in the study of transcription and translation regulation, and of course, we are limited to this setup that you see here now. So we have promoters, 5' UTRs, and almost always a coding sequence that we would like to either get the product out of it or regulate maybe in a cascade reaction. So it is an easy representation like this, promoter 5' UTR coding sequence. But of course, if you look at more into the details of it, then we can get a bit more branched. We can have all these function elements that are present within the sequence. In terms of the promoters, we have these very well-known 35 and -10 boxes. As we know, those are for the sigma 70. Sigma factor, and then we have many different regions that are categorized as upper elements. Some of these elements are core promoter elements that also spans across. Maybe one important to raise here is that promoter plus 5' UTR. Maybe I'll take the spotlight here. In terms of promoter 5' UTR, there are certain regions that are overlapping actually in terms of what the core promoter functions are needed. So this is a transcription start site. If this DNA to be transcribed, the mRNA will be starting here so that we end up getting with this 5' untranslated region. But again, the take-home message is, even though we look at the sequences in terms of promoter and 5' UTR, when we break them down to the function elements, there are many. And in my lab, we have the two focus on this approach. Bottom-up approaches in terms of understanding. I will exemplify how we do this. And another approach is in terms of controlling. There is more top-down, which we are moving towards by using the predictive transformer models. So the first approach that we came up with in terms of studying these regulatory regions was, if you're familiar with different approaches, if you want to target engineering promoters, you either take one promoter that you work with and giving all the function element that I have shown you so far, you modify one element at a time or certain elements at a time, or you fix one of these elements and need to do some random sequences in between, make libraries. Long story short, we have been doing this over a long time frame as well. The problem is that you're limiting the approach to only a single organism. If you work in E. coli, it's fine. You develop the sequence, which is also fine. But what do you do if you move to another organism? So how do you handle that? Well, you usually go back and take those promoters and do random, and testing their phenotype in these new organisms. So we were wondering what type of approach we could come up with so that we eliminate this type of, working with one host and then you cannot really transfer the information to the next one. And then the idea came, well, instead of we doing as well, fix the Shanagana sequence, modify the region, or take the -10, -35, modify in between or there. We said, I mean, can't we just modify the entire regulatory sequence? By that, I mean both promoter and 5' UTR, or in other words, all the region upstream of the coding sequence start. So that in their case would be the ATG. So it kind of experiment setup was like this. We ended up convincing different companies, and this story goes back to 2015, 2016, wanted to get companies to synthesize random stretch of sequences. Long story short, we ended up getting 200 nucleotides of random stretch of sequences, and then we put them in front of different coding sequences. These were beta-lactamase that gives ampicillin resistance, fluorescent proteins, different antibiotic markers, and even for Saccharomyces is a tryptophan pathway. This works in such a way that the Saccharomyces cerevisiae requires tryptophan to be produced and it lacks it. But if you were to provide in trans with a promoter on front, if it is functional, it leads to the growth of the cell. And using different genes, we targeted many organisms. This is E. coli, Pseudomonas, Thymus, Thermophilus, Corynebacterium, Streptomyces strains, and including, as I said, Baker's yeast. The approach is really simple. You take the 200 random nucleotide sequence with no premise at all. You just put it in front of the genes that you want to regulate, and you plate them out. If you're plating it out on antibiotic medium, and of course, within this library that you generate, and then the random sequence you put in, if any of those lead to viable colonies, that means that within that random sequence, in that particular clone, you have a functional promoter on 5' UTR. And with that work, we have built up the approach that in any organism, as long as you have some functional screening approach available, you can design, engineer promoters. What does it mean? You no longer need to rely on existing promoters. You can design them de novo, or not in a way design, but let the nature select it for you. You may also consider this one as going to accelerated evolution. And this is kind of the overview. So we made a library of, don't remember exactly the number, but in the range of 10 to 20,000 clones, not many. We sequence every one of them, and then you get a picture like this. So there is no hotspot. So this hotspots only is the GGAG, the Shandal-Garner sequence we've fixed within this 200 stretch of sequence. And then you do a lot of analysis. So we did DNA sequencing and RNA sequencing. And important thing that came out is that when we work with single promoter sequences, or we think that we're cloning a single promoter sequence, actually there might be that there are multiple transcription start sites in a way that you can have multiple promoter sequences in a given stretch. So this was a very interesting finding. We found up to 11 different promoters resting in just a single sequence, of course, occupying different regions of the sequence. And this was a very interesting approach. So it didn't only give us the option to design promoters or promoter and 5' UTR sequences, but it also discovered that actually you can have multiple transcription start site in a given stretch of sequence. So how do we take this work further? Even today's synthetic biology approaches, we are relying on pre-existing sequences. Even though we generate ourselves in this way, now we can expand the library of available functional sequences. Because I don't know how type of approaches that now listeners are using, but most of the time, if you're targeting a new organism, usually we take promoters that we worked in E. coli, and then you move on to these novel hosts. And here we have established this approach in Vibrio nat reagents using different fluorescent proteins. You take a sequence, random sequence, put it in front of the coding sequence, and then you generate a small set of libraries. Of course, the larger the library, the more variation you have, but so far, almost all the work we did, you get almost what you want. And based on that one, you can do a screening, either FACS or laborious hand approaches. In a way, you can identify functional promoter and 5' UTR sequences that leave you the phenotype that you want. And from there on is that you have a set of libraries. You can use them for different purposes. And I can tell that this approach also works with extremophiles, because it's agnostic. It's just a random DNA. As long as you have the functional screening available, you can use for whatever you want. And here, the orangish color are the identified promoter 5' UTR sequences that are functional in E. coli, and the blue ones are shown that work in Thermophilus. What this type of approach leads to is that you can identify sequences that are purely working in one host. For example, the one I'm pointing here, number 3, has very little, as good as no activities in E. coli. But if you look at on the other side here, these sequences are extremely active in E. coli, but have very little function in Thymus. And this has different applications. Another approach that we did with this approach, and with this technology or method, you might say, once you identify functional sequences, we pool them, and then you change the coding sequence to something that you're interested in. For instance, in the publication, we have included an enzyme and a carotenoid, and using thermophilus, we have showed that actually you can produce high levels of protein by simply creating a category of highly active promoter and UTR sequences. This is not the only approach you can do. Another thing that we are using this random sequence approach is determining sigma factor sequences or sigma factor DNA binding sequences in different microorganisms. In this case, I'm showing you, so the current project focuses on these 3 bacteria, Bacillus subtilis, E. coli, and Pseudomonas butida. In total, they have a really large pool of sigma factors, but we are targeting 17 of them. So what we did is that we take this random DNA sequences put it in front of an RNA aptamer called Mango. Mango is so that if the DNA is transcribed, it forms this loop where a fluorophore can bind. You can easily then detect in an in vitro transcription, given the setup you have, whether it leads to mRNA formation. And we're running in vitro transcription reactions where we add 1 sigma factor at a time, and then you pull the RNA that comes out of it using the random DNA sequence as a library. So that means if the sigma factor binds, it leads to mRNA that has to be a promoter. And in this way, you can also analyze sequences. It's a kind of length stretch due to time. I'm not going into details, but what you can do is we're generating millions of random DNA sequences. Libraries in this case is rather big. As I mentioned in the previous slides, we just made libraries in the range of 10 to 20,000. Whereas here we made really large libraries up to 1.5 million. And from there on, you can get functional sequences, and thereon you could do analysis, and it allows you to identify more than, in this case, 65,000 sigma 54 sequences that we have identified. Another important thing is, of course, with this approach, we're generating high quality and scale data, which we're also feeding into the model training. Yeah, maybe this is a cosmetic as well. We're doing the same thing in a microfluidic setup, because if you run things in bulk, you can't map the single template to the phenotype. Whereas in vitro transcription, we run in vitro transcription in the microfluidics droplets, and in that way we can identify single drops. They look like this, and if you isolate single droplets, that they contain only 1 template, and then with the intensity of the color we get, we can correlate or can generate this genotype-phenotype relationship. So as I said, one unique approach that we have in the lab that allows us to more get the understanding of transcriptional and translation regulation is we not work with model organisms, we work with a good stretch, and then we're applying different pools of these approaches to different organisms. Now, while starting from the random DNA approach, which allowed us to understand that there is a context dependency. Context dependency can be breaking down on a different level. So these are usually, it's in the DNA host and environment. What we have found out is that you can't take a promoter on its own, because promoter actually certain core elements that are represented at a 5' UTR. And then by the Lego approaches that we are using today, it kind of works and delivers as it does, but there is always these overlapping stretches of sequence that made us realize. I didn't share details of the result that we identified, but take-home message is these Lego type of approaches, it works. But if you take a promoter and combine with 5' UTR, then you're not reflecting on the unique characteristic of that unique promoter. Because if you take another 5' UTR, this initial transcribed region changes. And it's so on the CDS level, there is an overlap in terms of the structure and in terms of how the translation operates. Hence, by breaking these down, putting them in a Lego model, it kind of works, but of course it leads to a very unpredictable approach. Hence, when we wanted to do predictable DNA design approach, one of the take-home message from this random DNA finding was that we don't separate promoter and 5' UTR. When we design sequences, we consider them as a regulatory stretch. So in terms of using AI models for predictable DNA design, how we move on. So we took transformer models that are the same models that are behind the ChatGPT. Instead of training them human data set, we have trained them on biological data. These are usually omics data, proteomics, genomics, and DNA information. In terms of context, we can only handle DNA and host. So DNA context means the sequence composition and the regulatory sequence and the coding sequence has to match specific for the host. And then in terms of AI, so I said we are using transformer models, but in terms of design, we use promoter 5' UTR. and also consider the codon optimization on the CDS. And what we are predicting at the end, all of this is a transcription and translation rates. This is a bit of a lengthy verbose slide, but one of the limitations that we need to come in the field is that we are relying on existing characterized DNA parts. In a given organism, it may deliver as it does, but if you move on beyond a single organism, or if you won't move beyond a single gene, but a pathway you operate, then it becomes limiting. Another approach that we do is, that I will exemplify, when we do when we design promoter UTR sequences, we also don't consider on a single codon optimized sequence. We generate a large pool of that. And by doing an approach, we're not limiting ours to model, but we can actually go beyond a single host because the models are trained on a large number of data. And with this, we are enhancing predictability, and this just gives us the genotype-phenotype relationship. And the overall consequences of is that we don't need to design and test thousands of sequences or hundreds, depending on the scale, but we can just do experimentally validate just a few 10 to 20 sequences if the predictions are not too bad. So how does it work? The approach is simple. We define a CDS. I mean, obviously there is a gene that you want to express, or it could be a single gene, it could be a pathway as well. And then you define the host as well. And based on that, we generate not a single CDS, but a large number of variants. And for all these variants, we design promoter and 5' UTR sequence specifically, and then thereon we predict the transcription translation rates, and we rank them. Depending on the gene we're looking for, we select a few, and then goes to DNA synthesis. And then once you get the DNA, you do your expression studies. Here, a bit more, maybe meat on the bone, but these are real numbers from an experiment, actually, that we have delivered. So this was a Mulvarti. This is reverse transcriptase used heavily in both in diagnostics and therapeutics, and actually during the COVID times as well. So this is currently produced by using E. coli in the line that I'm showing here. So we threw this approach to our models to say, giving the construct, this is already produced enzyme, we already have the DNA construct, we know how much it leads to. Can we improve this? So the approach was as following. We took the coding sequence for this gene, we codon optimized it, and at that process with parameters, of course, you should not have the sequence, it should not have this and that, with constraints, and then about 100 codon optimized variants. For every one of them, we generated promoter and 5' UTR sequences, and that took the number up to 11,000. From there on, we predicted transcription translation rates, and then we ended up simply taking 5 sequences and then validating them experimentally. And of the 5, these 2 worked very well. Depending on how you look at the soluble, insoluble, the production could be increased 40 to 70%. And this whole setup took simply 4 months to complete. So what does it mean? That means by using instead of relying on random approaches or always taking one single promoter and trying to express your gene, we could use predictive design models to reduce the number of experiments that we need to perform. So it's practically the way we are operating is it reduces the experimentation. Okay, I think I'm hopping over to a bit kind of the details due to the time. Just to come to it, so these sequences, these are just kind of experimentally validated sequences that are purely de novo design. So these are designed specifically for the host. In this case, this was a fluorescent protein. And the reason I'm showing you the sequence is that there is no grammar in a language sense behind the predictive models. These are simply trained on large omics data set, and they all can identify the context specificity giving the CDS and the regulatory sequence. And as you can see, it also suitably puts the Shanagana sequence in the right distance and in what the host prefers. And this is just the output here on the right is the BLAST result. There is no significant similarity found, so that means they are truly de novo, in a meaning that it's not takes a known sequences, introduce some mutations here and there to give a novel one. So these are truly novel. Even if you choose the low similarity, you still not get any similarity. How are we using this approach? The details are long in this case, but if you were to clone a single gene with a single regulatory sequence, you do cloning, ligation, and then your transformation. Instead of doing that one, we maybe increase the number to 20 constructs. But in that way, in a single project, we're targeting by using different organisms, different coding variants, and maybe different expression levels, you kind of generate this a different solution space with every construct ending up being here and there. in a nutshell, what it means is if you were to construct single cassettes and then test their phenotype in a large solution space, so this is a large solution space, you're occupying one area. So by using this approach, then we can target multiple designs, multiple hosts, and preferably different coding variants. And then in one shot, you're getting a much larger solution space covered, and then you can pick up whatever that is best for the project and go on with that construct. So up to now, I have only talked about things that we have designed and built based on our data, the way we curated data, and etc. So the final validation was we took data from the literature. The models have never seen them before. In this particular case, this is from Nico Klassen's group in Wageningen. So they have designed, they were testing, I guess, different coding sequence optimization algorithms. And then they have characterized meticulously almost 1,500 constructs. So these have fixed promoter and 5' UTR sequence, but different coding sequence variants for the red fluorescent protein. And these are depicted in the blue line. So some of them are really poorly performing, some of them really well. We took the sequence from Nico and then threw into algorithms that, if you were to predict the sequences of all these 1,500, what the model thinks that are the best performing ones, those are the red ones. So what does it tell us? Why? Once it tells that actually it's not 100% accuracy. And actually maybe if you put that on the R scores, maybe 0.6 level. But what it allows you is that you don't have to have the 1,500 constructs. So if you were to try to get some constructs that are performing really well, then you could easily start with one of them. You're already halfway there, because once you have a working construct, you can improve it. And the models are agnostic, and this is an experimental demonstration that it works for the same similar data set for Vibrio and a similar data set from Streptomyces, because we trained on Ensemble 52, I think at that time, at that more than 4,000 genome sequences. We are covering a good range of microorganisms available. I think it's 25 now, but maybe I should take the last thing. One thing that you may wonder, how the algorithms are working. On a technical level, we have an understanding, but we have no understanding on what the DNA sequence function level, what they do understand. In order to decipher, or we're not totally understanding it, but to kind of tackle how do they understand, what we are doing is we're giving a sequences, we predict the outputs, and these are experimentally validated as well, so predicted here and true labels here. And then we're using network analysis to see this importance or the significance of the sequences within the DNA. And then we generate different variants based on the weights. These are marked as green, meaning that they're adding improved performance. But if they are red, they are pulling the score down. By generating these variants, then we can create constructs that are performing well. And then we use our current understanding of biology to reflect on, okay, maybe this and that is happening. But long story short, actually, we cannot always nail down the single parameter. So one of the things that we came up by these approaches, that in my academic profile as well, I would like to take one parameter at a time and then change that parameter and then have a bit of understanding. That would be, only focus on secondary structure or the spacing between the -10, -35. One thing that we have done is that actually we moved from that thinking. We're targeting as many parameters as we can because life is short. And then this gives a bit of a different perspective, is that actually we don't necessarily need to understand what the function is, as long as the DNA that what we're working on can give us what we are interested. So maybe that's a bit of kind of reflection on the work. With all these features, so what we are doing is now we can engineer promoter and UTR sequences. We do this from a single gene. We could stretch this on a pathway. The time is running out now. I don't have experiments. I couldn't demonstrate it, but we're doing it for pathway engineering. And another aspect is that we're not limiting ourselves to a single codon optimized variant. We actually generate a large number and then handle them all, because there is no winning codon optimization algorithm. And then we are also not limiting this type of approaches to single hosts. Another thing that we are doing is in the development, of course, with this, you can also do protein engineering, not just engineer the sequences, but you also consider then the function of it. If you're a bit interested to hear more of the details, this is the preprint that we have submitted, so feel free to read and of course reach out and ask at the end. Yeah, so this is what I was coming to tell, that it was not just simply on that we need to understand, rationalize our design, but actually we can let the AI to do the design as long as it gives us the function. So there's a quite large number of people involved from my lab and NTNU. And we have also a spin-off that is handling this technology right now. I'm adapted to our collaborators in University of Bielefeld, who are actually generating both the DNA and RNA sequences, and of course, a bunch of different funding bodies. Well, with that, I think I went a little longer than 20 minutes I intended, but I am happy to answer the question that you should have.
Well, that's perfectly fine. Thank you very much, Rami. That was a great talk. And I think, I mean, I don't know about the others, but I have like 1,000 questions. So, but I'm just gonna ask one and then open the floor for more questions.
Yeah, please do so.
So it's not just me. So yeah, just to check if I understood correctly, you're generating this N200 libraries, and then from those candidates, about like you get like around like 12,000 candidates, and then from those, you pick some, you describe those, and then you use the sort of that information to train your AI algorithms.
No, sorry, that must have given a wrong impression. So the way we do in this one has nothing, it has not been used. We only did it for validation, but we haven't used them in training. Because in the training, I mean, of course, you could use it in training as well, but the scale of data we get when we parse genome sequences, it's beyond what the numbers are here. I mean, we have characterized so far maybe close to 1,000 sequences. Just in a single genome, you get like more than 3,000 or 4,000 promoter sequences, right?
So then if maybe, I don't know if I should ask or not, but like for training, so like the data that you use is data that is like already available, or how do you train your AI data sets?
So those are, let me see, I think I had a slide on that one. So these are the public data sets we use: Ensemble Bacteria Release 52, and a good chunk of RNA-seq data, both from online repositories from our network, as I said, with University of Bielefeld. Another thing that we have also did, we curated and compiled quite a bit of data from the literature. So it's a combination of all this that went into the training, and of course, a bit of proteomics and protein embeddings as well.
Yeah, that is fantastic. So I guess my question would be, are you sort of like, is there in any of your prospects using some of the new data you're generating to try and train data sets? Or
yes,
because,
so
yeah.
In the Sigma project, like in the project where we are generating the let's see, the Sigma sequences, because the thing is, so in terms in AI world, there is two approach. It's called supervised or unsupervised. So in the unsupervised fashion, as long as you have DNA sequences and RNA sequences, the algorithms generate this correlation between, what type of DNA sequence comes in, what fraction of those are functional, to what extent, and then that generates the correlation, right? I mean, that's actually, AI sounds fancy, but all they do is this correlation, but of course they could do this in a massive scale. But what we are doing now, because once these sequences are generated, like I was telling you, we're getting these functional sequences, but we have no idea, how are they regulated, what regulation applies to these. So now we are generating data by using this random DNA approach, but of course not purely putting into the cells, but rather running in vitro transcription reactions in the presence of sigma factors. We are able to identify the binding sequences. With that data, right now we are training models. With hopefully offering a free server for promoter predictions. That's the only data we use currently that goes into model training the models. But that is not going to improve our models. It's just going to be reflecting on what type of regulation might apply to those sequences that the models are generating.
Perfect. Well, thank you very much.
No
So I
problem.
would like to open the floor as well for other questions, so that I just don't continue myself just asking 1,000.
Yeah, no problem.
I have a question if no one else has a question. So very interesting. I also work at the intersection between models and non-model organisms. What if someone has, let's say, a library of riboswitches that they would like to test in the 5' UTR? How could your method be applied to some application like that? Sorry, we can't hear you.
Rami, I think you're muted.
I'm sorry. In the random sequence approach, you can fix any sequence you want. So if you're using ribose switches, you can limit them. You can either create a set of libraries of your ribose switches, or you can literally use one of them. Or you could take a combination of that, fix certain areas and modify the areas around. And then you could, for instance, use random sequences upstream of the promoter, and then screen your library. And of course, you have to create a library first, and then you have to decide how large a library you want. By the way, one thing I've forgotten to mention is this approach. So if you use random DNA upstream of coding sequence, about 30% of these sequences are functional, which is a it was unexpected finding for us. So if you make a library of, let's say, for simplicity's sake, 100 clones, 30 of those clones will give you one or another level of expression. And then you can combine it, any sequences you fix. So one thing that I haven't mentioned, for example, or maybe I did, I guess. So we either kept it totally random upstream, or we fixed the Chandra sequence. And actually, irrespective of that one, of course, if you fix the Chandra sequence, the translation is kind of half taken care. So you get a little bit higher than 30%, but it was not close to 40. But nevertheless, so this type of approaches are totally doable. I don't know if that answers your question. Was it Rosa you asked?
Really interesting. Yeah, thank you.
But it's more, I mean, I think the idea here is using this approach is to generate data that you can go back and analyze what's going on. So you're giving the nature the possibility to tell you what's working or not. Because one of the limitations in our approaches, or standard approaches, like people still do, fix the 10 minus 10 minus 35. But why are you fixing it? Because 30% of this library, it generates practically functional sequences. And that way you can generate a much richer data. And that also goes back to, you don't study one parameter at a time. Because if you fix minus 10 minus 35, that means you're going to use sigma 70 only. So that means any sequence you're going to generate will be sigma 70 regulated. But if you want to generate breadth of data and then hopefully gives a bit more in-depth understanding, then you can go a little bit more free. And I think that's what I like about the approach. So not targeting single parameter, but multiple parameters at a time. More questions?
I would like to ask you a question, but on that reflection, because I'm very interested in specifically kind of the idea of what you do in terms of working in the intersection of like wet lab work and computer work or prediction algorithms. My question would be, because you basically generate libraries and having selection platforms, and then you're also training AI and then testing that, like what do you find the most complex part of the process? Do you struggle more with the wet lab? Do you struggle more with training the algorithms? It sounds like this
DNA synthesis.
The N200, that'll be the issue.
No. Sorry. I mean, okay. So in the frame of the work that I explained, the most painful part of is getting the sequences. For instance, we wanted to use the 200 in vivo as well. But the DNA synthesis could not overcome the contamination of ribosomal RNA, for instance. And different people try with different approaches. I don't know why. Maybe there is something inherently problematic with this random DNA approach. So if I have to tell, that was the limitation for us. Otherwise, it's pretty straightforward. Making libraries, it's painful because you need, transformation efficiency is, you can improve it so much. I think at best we get maybe close to 100,000. So if you want to make a library of 1 million, then you need to make 10 transformations, quite large number of plates. These are manageable. Pool those sequences, send for synthesis sequencing. That was also pretty straightforward. So that bit is really, there is not much can go wrong. What goes wrong is that if you have some design ideas you want to test, we usually don't get all the sequences we want to work with synthesized. But in terms of if you want to, apply this to different organisms, also we are helping our collaborators. Really easy. There is no challenge really. But the limitation on the predictive approach, when your predictive approach leads to certain sequences, we also ditch some of those because we never get them synthesized for technicalities. But that being said, nowadays we are moving to a recent company. I mean, I can't do advertisement now here, but they do a different approach. And then hopefully they claim that they can target much larger sequences with high accuracy. So if they resolve the problem, maybe that will ease us in the future, but currently I don't know. Otherwise, we work with companies like, GeneScript and Twist and all that.
That's very interesting. Thank you very much. So I think if there is any other question, this will be the moment to ask it. But if not, then Rahmi, I would like to thank you very much for your time and for this great talk. It was very interesting. And thank
My
you also
pleasure.
everyone for participating of this. So I guess I'll be seeing you in the next seminar.
Bye for now.
Bye. Thank you, Rahmi.